Hello.
Hi.
How are you?
Can you hear me?
Yes.
Yeah, oh, okay. No, well, I'm here.
Yeah, good.
I don't know about anybody else. Um,
Hopefully.
yeah, I think I can actually see just as well without the glasses.
Oh, yeah. Well, that's
So
good.
maybe I'll...
Yeah. How have you been doing recently? Do you have any updates?
Myself?
Yeah.
Um, no, I'm recovering from eye surgery, so I don't know if you can see
Yeah,
that.
yeah.
So my reading ability's gone downhill.
Oh,
Like, I can read without my glasses on, but I'm sort of like out here somewhere.
yeah.
I need reading glasses. Anyways, yeah, so I've been taking it easy, sort of.
Yeah.
For 2 weeks. It'll be, if I do this for 3 weeks, then things will have recovered enough that I should be back on board.
Yeah. Well, that's good. I guess it's the summer, so you get like there's lower expectations for your graduate program
Oh, I'm supposed to be doing a major project.
right now, or like.
Yeah, absolutely. And I'm not doing it, so it's not really a good thing.
Well, yeah, that's
I
true.
Yeah, I found a few papers that were very interesting. Um, I could pass them on to you, I guess.
Yeah, that'd be good.
One, yeah, one's about electricity, yeah, and how um, nerve cells automatically create a voltage.
Okay. Yeah.
Anyway, so
Yeah, yeah. You have to
I'm
send them along. Yeah.
okay. Yeah, I sent them to Dick, and I don't know if he ever sent them to you. So
Okay.
nope.
Yeah.
Okay, well, I'll
I.
send them.
Yeah. I'll see.
Well, it's interesting because long cells, like nerve cells, seem to create a voltage, and also long cells, as in heart cells, seem to create a voltage, and also just muscle cells in general.
Right.
So I don't know.
Yeah. Well, that should be interesting to look at.
Yeah.
Yeah. Harikrishna, Karan, how are you?
Hi, Bradley. Hi,
Hi.
Susan. Yeah, things are going okay. I'm still working on the projection method part currently.
Okay.
And since the midterm evaluation is due, I'll be writing a blog post related to that as well, detailing all the work that we've done.
All right.
Other than that, I think this for week 7 from July 25th till 31st, it's mostly just categorizing whatever we've done and putting it properly under a template with proper documentation. So this is what we planned for this week.
Right.
Otherwise, week 6 is work the week before this, which was this is still going on. I'll still keep on. I'm still looking for a better fit for the projection method as of now. Yeah,
Yeah. Okay.
that's the major thing that's going on. Otherwise, apart from the projection method, the model is okay. I've not done too many changes after that. Yeah.
All right. Uh, Looking ahead, is there anything you can do, sort of, as you're searching for the better method? I mean, are there other things that you're kind of moving forward with?
Yeah. Like earlier, I was using there are two algorithms that try to find the corresponding points in two images, two adjacent images that are there. So it tries to find the matching areas so as to identify how much they rotated, as well as identifying features and all, called SIFT, SOF. So the thing is, OpenCV used to have these algorithms, but since Google has patented them, so it's kind of like in a third-party area. pattern wise. So there are like alternatives for this. So I'll currently thinking of going through ORB as another alternative, to replace this. Otherwise, this was again something I was looking into, and the projection method, of course, is another thing I'll be doing this week as well.
Okay.
That is carrying on from the previous week.
Yeah.
So Google Summer of Code won't let you use Google licenses? What's up with that?
No, it's like the Python library that I was using, OpenCV,
Yeah.
used to support using these algorithms with the library itself. So I didn't have to hard code or create an algorithm for each specific case on my own. They used to have boilerplate code that used to do it for you. So in the recent versions of OpenCV, this thing is not there. They've removed the algorithm that used to do all this processing on their own end. So I'm looking into alternatives for this. Otherwise, I can use SIFT and SOF. It's just I think just for the project's sake, I was looking through going through some different options, because the OpenCV libraries dependencies, that will be included in the final project could affect somebody in the future, who might think of using this, because this feature won't be there in the newer versions. So
Right.
on those aspects.
Why did they remove this?
SIFT and SOF, because I think it was something regarding the licensing of open source projects. So they can't keep it open source. It's I don't know. The reason for this is kind of like, even I don't know it properly, but the main gist of it was that since the engineers at Google had come up with the idea, and Google usually gives away everything for open source. It's just they the OpenCV reason that the OpenCV guys were citing was this only.
Yeah, it's usually some sort of, they want some intellectual property out of it, so they don't want to put it in that package. Sometimes they also, I don't know who runs OpenCV, but sometimes they, terms of use get a little complicated, so I don't know.
What would the SIFTs of algorithms that are there, and them not being there in the newer versions of OpenCV, and ORB as a replacement that I'll be trying out within this week, along with the projection method improvements that have to take place to get a better model fit.
Yeah.
These are the major updates for this week.
Okay. Yeah. I don't know. I don't know if you caught my question, but is there a way do you have any references on the SIFT-SOF algorithm that we can, like a Wikipedia page or something, that we can review? I know you're not using it, but would you mind, is there like a Wikipedia page for like the SIFT-SOF algorithm? We could look at it, and because you're going in that direction.
I'll just share the link. Yeah.
Okay.
OpenCV.
Yeah.
I'll just do it. Yeah.
Okay. All right. Thank you. Harikrishna, do you have an update?
Yeah, hello.
Hello.
Yeah, so in the last week I told that I had collected those 3D coordinates in a file. The extension is called .ply. So right now I have managed to load that file into my JavaScript code and display it in the application. So I am able to view those point cloud now. So right now what I have to do is create mesh from that point clouds. Then I'll be working on the projection methods. Then as the mid evaluation is coming, I'll also be documenting the things down to like what I've done till now.
Alright.
Yeah, that's what I've done.
Okay, sounds good. What are your plans for next week?
Now what I want to do is project the images onto the mesh which I've got now. So I'll be working on the projection methods. I'm not sure what I will go ahead with, but I'll be experimenting 2 or 3 projection methods and choosing any of them.
All right, that's good. You were having some technical difficulties before, like with trying to get some things to work.
But I had mentioned some difficulties before, but I told that I had solved it.
Okay, good. Yeah.
It's solved.
All right, good. So looking forward, do you anticipate any sort of difficulties or like?
No, right now I don't see any difficulty which I might face.
Okay, well that's good. Yeah. So this week we'll be. Thank you for the update. So this week I'm going to be doing the evaluations for GSoC, and we have 4 people. We have Harikrishna, Karan, Wataru, and Jiahong, and I've been talking with Wataru and Jiahong. I've been keeping updated on Slack. So I think all of you are going to pass, and I just need to write up the evaluation, and you'll be getting copies of that later. But I mean, like, I like Karan's idea of writing up a blog post on that, an update. You're not required to do that, but it would be nice to kind of reevaluate your project at every evaluation step, just to make sure that it's still within scope. That's all I was asking about, what you plan to do looking forward. I wanted to make sure that we're keeping on with the schedule, and we have, I guess, one of the things they asked me was that if people need extensions, we can request them. So typically the program runs, I think, 12 weeks, and then, you get your evaluation. But if you want to extend it, if you need to, submit it later, maybe in the fall, I think they allow you to extend it up to 22 weeks. So that's pretty flexible. I can request an extension, and then you have a new due date. So I don't know if anyone in this group will be requesting an extension. We have one of the shorter projects. So the longer projects usually, I think, are going to get the extension given more freely. And we have, in my other group, we have a longer project time. So I don't know how relevant that is, but if you do need an extension, please let me know. Okay. Just wanted to go over, actually Morgan's here. Hello, Morgan. There's Karan's link.
Oh, yeah.
Oh, thank you for the link, Karan. I just wanted to get an update from Morgan.
I just have a clarification regarding the SIFSO thing. So yeah, it's been discontinued in OpenCV, but it's not. I think it's not. It has been patented by its own creators, the people who created the algorithm. So it's there for free, and academic use and research settings. It's free for that, I think.
Okay.
But the person who will be using the library, he'll face some issues, I think. That's the only.
Right. It usually has to be supported within OpenCV.
Yeah. So the OpenCV part supporting it is not there. Otherwise, I think if you can make do with other things that are there. Orb is there, which is slightly better. It's mentioned in this article as well.
Okay.
So in the notebook, yeah.
Okay. Yeah, I'll get an update from Morgan, then we'll go over that a little bit. So, Morgan, hello.
Yeah, hi, Karan.
Hi.
Yeah, not much of an update. Just listening and caught your embryology video, which still processing, but
okay.
But that was great, and yeah.
All right. Yeah, good. Nice to hear. Nice to have you be able to make this meeting. So, yeah. So I do want to go over some things here. The first one is this article that Karan, it's, let me share my screen here. So this is Shubaka 89 on GitHub. This is OpenCV with Python. this is the SIFT, SURF, FAST, and BRIEF and ORB repository. So these are the, I guess, the methods that they're using here. So this is a feature detection algorithm. The SIFT and SURF algorithms are patented and free to use in academic settings. So, and then, so this is the SIFT algorithm here. And this has, the code is here. And then there's a link to the, I guess it's some documentation on it. So SIFT stands for the Scale Invariant Feature Transform. So this is a lecture on it, kind of goes over some of the things. And if you go into the link, you can access this stuff. So fundamental to the many of the core vision problems, recognition, motion tracking, multi-view geometry, local features are the key. And you have detectors and descriptors. Yeah, so this is basically the structure of SIFT. Find scale space extrema, key point localization and filtering, improve key points and throw out the bad ones. Then the descriptor is orientation assignment, remove effects of rotation and scale, and create descriptor using histograms of orientations. And then detector is, okay, so they go over that again. So this is a basketball. They need to find characteristic scale for feature, and then the scale space. And then this kind of shows, I guess, how they take something and they turn it into a sphere. Is that what they're doing here?
Yeah. It's basically when you have two different views of an object, be it views from a distance, two different distances, or from two different angles. It helps you in finding the key points that are there, and using this algorithm, I'm using it for finding the angle of rotation and 2-3 more other things, because I know the key points that are there, how much they've moved, and for these are the 2-3 functions that I'm using for generating the model, the amount that I have to rotate the outlines as well, these sort of things.
Yeah.
So in that blog only, he's mentioned a better way to go about these things, because these things take time. So ORB is currently the most recent algorithm, I think, for doing something similar. So SIFT is there, then SURF is there. After that, the next best thing is ORB. So I'm thinking of even incorporating that as well. Otherwise, the main useful aspect of this algorithm was the key point detection and finding the displacement of points and all those things.
Yeah. I'm just making a note here, just to think about it. So yeah, you do have a sort of a alternate version or some alternate method that you're going to use, but it's going to basically do a similar thing.
Yeah.
Okay.
As a replacement.
Yeah. And then, so this is the SIFT method, and then the SURF method is here, the FAST method, the BRIEF method, and then this is the ORB, orientated, fast, and rotated brief. So this is where you're taking, they don't have it up here. So this is where you're taking this SIFT model, or no, they're taking the fast model and the brief model, and they're turning it into this ORB model. And they don't have a document here, but this is the code. So it's using very similar things, key points, descriptors, and then it's turning it into that sort of projection. So that's nice. That's interesting. Thank you for that, sharing that link. In terms of pull requests on DevoLearn, we have Wataru's been making some pull requests here for different things. We have, we haven't done a lot of them lately, but he's made, I think, one other pull request this summer code. So he's working on the GNNs project, and he's working on importing data and getting that pipeline set up. So he's working on he was having some problems with the cell segmenter, and he's been improving upon that. So congratulations to Wataru for that. also, I've talked about the mathematics of DevoWorm in past meetings, and so I finally got this up and like into a poster form, and it's going to be presented at Find Your Inner Modeler, which is an NSF sponsored workshop. We've had about 5 of them so far, so this is the 5th one, and it's a nice event. It brings together biologists and modelers and brings them together in one conference. And then the conference setting, they basically try to find different ways for them to talk to one another. So, biologists will give a talk on their biology. The modelers will give a talk on their modeling. Sometimes you have a lot of people with both sets of expertise. So, they give talks and they kind of put the biology in the context of modeling. So this is the Mathematics of DevoWorm poster for this particular event. And, I've tried to take some of the parts that I've shown in previous meetings. Let me see if I can get this at a better size here. All right. And add some biological context, some modeling context. And then this is what's going to be in. So, basically in a poster, you want to be very visual. So all of these different points here that are labeled with letters, these are all from papers that we've done in the group at various points in the past. So, we have these different things that, we want to do things to characterize development, different aspects of development. And we have some models that we've proposed, we've written about. And so that's what this poster is about. It just kind of goes through those different models for different things. So the first one is, how do we capture the multitude of interactions and state changes, or cell, which could be like cell identity or other things that occur across embryogenesis. And in this case, we're thinking of discrete dynamical and categorical models. So in this case, we have a category theory model that was in one paper from 2021, and a developmental game theory model that was proposed in 2018 in another paper. This one is, how do we capture pattern formation and cell migration dynamics as a computational process? And our answer is cellular automata and spatial data structures. So we have some examples here. We have an example of a cellular automata producing a sort of a modeled pattern or a dotted pattern. And so this is, pattern formation in a cellular automata. But then we have these different models that you need to underlie that, different computational representations. So we have the von Neumann neighborhood, which is used in cellular automata. We have this 5-tuple data structure where we can take data from cell tracking or some other source and create this volumetric representation. So XYZ, and then time, and then this division angle, which is something that we can derive. And we've talked about this in our graph neural networks. This volume can also be extracted from some of the work that we're doing on projecting to a sphere. So this is also something that can be, and then the angle might be like the angle at which it's projected to. I mean, there are different ways we can do this, but this is the sort of the core representation here. And then there are these other things like, well, actually, this is a different question. But this, anyways, this is the C. And then this one is D, which is, how do we characterize the organization of developmental cells, expanding morphological structures, and emerging connectomes? And our answer is neural networks, complex networks, and trees. So every time we ask a question, we give an answer. And the answer that's in bold is our group's answer to that. Not necessarily like the definitive answer, but it's just one possible answer. People can do this in different ways. So this is actually out of place a little bit here. All right. So this, if we go to the diagrams here, this includes node attachment and complex networks. So this is where we're growing networks from maybe 2 nodes to 50 nodes or 500 nodes. We have to add in nodes every time, we have some growth event. So if something's growing, if it's adding nodes, if cells are dividing, or if our scope is expanding, or if the shape is changing, then we add nodes, and we have to have some sort of attachment process for that. We also work with neural networks. So neural networks are interesting because although they're different types of networks, so they're not like complex networks, they still need different rules to have. This is really not okay. There we go. They still need different rules, and we use these to model the nervous system, perhaps, of a worm instead of the morphology. So we use it in a different place. Let me just get rid of that. Then we have lineage trees, and lineage trees are, of course, ways you can trace a lineage of an embryo. So you can trace the individual cells as they produce daughter cells. As the cells differentiate, we can characterize those with the lineage tree. So that's very nice. And if we go up to the top here, we have lineage tree dynamics. So we've also done some papers on lineage tree dynamics. This was from a paper in 2016, I believe. This is a computational study on looking at what happens when you flip the nodes at a particular level of the lineage tree. And you also, if you represent it as if you represent it as a differentiation tree, it looks different than if you represent it as a lineage tree. In C. elegans, it's really about ordering the nodes from smallest to largest instead of the thinking in terms of pure heredity. So you have two alternate trees. You have a differentiation tree and you have a lineage tree. Then what you can do is you can create random conditions. So you can create trees with nodes that are just jumbled around. So there's no heredity and there's no other criterion except just that the nodes are randomized at each level of the lineage tree. Then you can compare those two and see what their information is. And there's an information measure in the paper that we use. And you see that in C. elegans, when you compare like biological trees, which are the ones that have some ordering criterion, to random trees, that there's this difference in terms of their information, or we use Hamming distance in this paper. But basically there's this greater amount of information in the tree. And so it's kind of an interesting study, but I don't want to get any more into it because it's one of these things that's hard to describe any more than that without reading the paper. So this is an example of a lineage tree, and this shows you the mapping between the lineage tree and the number of cells in an embryo. So this is from someone's paper here. I just use this as an example of a lineage tree. But then this paper was from 2021. This was on looking at simulating cell divisions using a number of distributions. So that shows that certain distributions are provide you with different patterns than others. In biological cases, it looks more like this yellow function than the other functions. So there's it's kind of following up on this work, except it's in a little bit different way. But anyways, we've been interested in this idea of lineage tree dynamics. And the question is how do we characterize the rate, recombination, and conservation of growth and form? And the answer is lineage and differentiation tree modeling. So we've done this sort of work, and then this is something that's emerging where we're using agents and looking at like building these differentiation trees where you can build agents that look like what we call Bratenberg vehicles. But you can use that to build these vehicles, build these complex phenotypes, and trace out the differentiation tree. So you're using different types of what we could call tissues, which are just different parts of the vehicle, and then you're letting it differentiate and divide into new parts. And you're tracing all of that through one of these lineage or differentiation trees. And then, so that's the end. And then at the end, we have these references, which are the different references that we're using in our group. And then at the top, there's a guide to the link for publications on the group website, so people can get a handle on some of these other things that are going on in the group. We have someone else new here. Longhui, hello?
Oh yeah.
Hi, how are you?
Hi. Sorry for late for lating.
Oh, it's fine. So, how's the Graph Neural Networks project going?
Yeah. The stage one source code is releasing, and I use it to process the video. Yeah. And then I was studying the results. Yeah.
Okay. So you're at stage 2 then, or
Pardon?
you're at stage 2 right now? So stage 1 is pretty much complete, or?
Yeah. Stage one is going. Yeah.
Okay. So
And stage two is Jahan's work.
Yeah. But you're coming along on it. It's
Yeah.
okay. Good. Yeah, that's good. So that's good. That's all making progress. It's pretty ambitious, but I think it's doable. We can get it done. It can be done. So that's good. And I saw that Wataru had posted some pull requests for DevoLearn. Okay, so that's good. Do you know what Jiahong and Wataru? Do you know if how they're making progress, or? Are they running it? Yeah.
I don't get in touch with him. Yeah, I just work in my way. Yeah.
Okay. Yeah, I mean, I didn't know if anyone was running into any problems, I guess. If there's any outstanding issues that need to be addressed, or.
Because the source code is great, so I just can study it well. Yeah.
Okay. Well, that's good. Yeah, thanks for the update. And I'll be in touch on Slack periodically. So, all right. Thank you. Did we have any questions about anything that we talked about in that previous section? Okay. Well, we have I want to get into some papers now, if you don't mind. I have some. Last week I recorded an update in addition to people's GSoC updates, and I put it into the video that was on YouTube. And there are two topics that were covered. One is a really interesting set of papers on sort of an alternative to reaction diffusion morphogenesis, and the other was on critical periods, light exposure and critical periods in chicks, which is interesting. I mean, it's usually, very behavioral, but it's also, it's part of development. So this week I'm going to go through two sets of papers. So the first one is, and you might find this interesting in terms of systems biology. I know a lot of you are maybe familiar with systems biology, but if you're going to be doing computational modeling, this is important to know how this, at least be aware of this kind of work. So systems biology is really an area that, where they do focus a lot on genomic data and things like that, but it's really about, making biology, turning it into computational artifacts or computational methods. And so a lot of this work has been on high throughput data, like gene expression data or genomic data of some type. But still, there's this whole field of systems biology. People are also interested in physiology, like measuring physiology in real time, or proteomics or things like that. So it's pretty diverse. So this paper is on modeling formalisms in systems biology, and this is out of AMB Express. And I'm not really familiar with that journal, but anyways, the abstract reads, systems biology has taken advantage of computational tools and high-throughput experimental data to model several biological processes. These include signaling, which is like chemical signaling between cells or signaling within cells, gene regulatory, and metabolic networks. So gene regulatory networks are these networks of genes that produce some sort of gene product or gene expression. So mRNA, or even the role in producing proteins, but in an indirect way. And then metabolic networks. So metabolic networks are where they look at metabolites and they look at other types of, or they're really vast networks that they model, and they're chemical processes in the cell. So you think about things like ATP and other types of energy production and other types of things like that. That's what they're doing with that. Gene regulatory networks are, of course, they give you some readout of maybe like mRNA, and you can look at the fluctuations both in at a single time point and over time. And oftentimes you'll find things like different types of fluctuations, different types of rhythms, and so it's interesting. I mean, most people just measure gene expression with relation to like an experimental condition. Like if you take out, if you treat it with a drug and these cells, respond in kind, and you take your measurement of gene expression and you say this is, it's upregulated or downregulated as compared to something that didn't experience the chemical treatment. But you can also look at gene regulatory networks over time, and in fact, that's where I think a lot of the information there lies. So there are all sorts of, they focus on networks, but systems biology features a lot of other types of structures, and such as like putting the data into an atlas, mapping it to tissues, and other types of things. And the term systems is very loose. If we talk about systems, usually we talk about more than one part. So it's really about a lot of parts interacting. So you can use networks, but you can use other things as well. However, in the field of systems biology, most of the models that they use are specific to each kind of network. So your networks are, they use a certain type of network for a certain type of problem. There aren't really network models that bridge every area of the field. So if you're doing metabolic network modeling, for example, you'll use a certain type of network protocol. You'll use like a certain type of, set of rules. And then if you move to gene regulatory networks, you use a different set of rules and a different network structure. And if you think about it, that can be very difficult to reconcile. So, if I take a systems view of gene regulation and I take a systems view of metabolomics, which is what they call metabolic, the study of metabolism, I can't really reconcile those networks. It's hard to bring them together and say, this is happening in gene expression, this is happening in metabolomics, and here is the connection. It's very hard to do. So but these systems do interconnect, and really that's also part of the systems approach. But because of the way these systems or these network types have been developed, it's very hard to make that connection. So in this paper, they describe the features required by an integrated framework for modeling, analyzing, and simulating biological processes. In this paper, they review several modeling formalisms that have been used in systems biology, including Boolean networks, which are these networks where the nodes have two states and they switch between the states. Bayesian networks, where they have a predictive aspect to the network that uses sort of the likelihood of maybe a connection or maybe the next state to be reached. Petri nets, which are kind of mysterious to me. I've never understood them, but it's a different type, very different type of network modeling approach. Process algebras, constraint-based models, differential equations, rule-based models, interacting state machines, cellular automata, and agent-based models. So that's a pretty broad swath, and they're going to review all of these methods. And really you think about these different methods, and they sort of do different things. So, you have your differential equations, they're modeling some sort of time series, they're modeling a system that's unfolding in time. Whereas your, agent-based models do the same thing, but they do it in a very different way. They do it, without explicitly referring to equations. They might have an equation as a rule, but they don't operate in the same way, or that you can't analyze them in the same way. And so they compare the features by providing different formalisms and discuss recent approaches in the integration of these formalisms, as well as possible directions for the future. So it's kind of a longish paper. It kind of goes over a lot of these methods. I can put this in the channel on the Slack if you're interested in reading more about it. I'm not going to get deeply into it, but I'm just going to go over through here and see if there are any nice takeaways to bring up. So, they get into this idea of biological networks. You have these different examples. So here's an example. A signaling network is on the left, where you have this sort of topology and you have these symbols. And, you're looking at a very specific set of relationships. You're looking at signaling, from one component to another. Then you go to gene regulatory networks, where you have a very different structure. You have your genes, you have your products, you have your promoters and your enhancers and all that, and then it's producing something, some sort of product, some mRNA or something like that. And then you have metabolic networks, which are these metabolites that are connected together, and there are different rates and their rate limiting factors, and so there's these dynamics inside these networks that are really interesting. But the key problem here is that you have a difficulty sort of reconciling these networks. So, for example, these green boxes are proteins in this model, and the proteins in, say, a signaling model might connect to regulation of a gene in a gene regulatory model. And so we can map that, but it's very hard in practice to actually calibrate the model with data, because we can, say, follow these signaling pathways to the production of proteins, and then we can follow it to a gene regulatory model. But the data sets often don't let us, necessarily just plug in the numbers. Furthermore, these models are going to be, very different. They're going to become very complex, and they have different sort of conventions. So signaling pathway producing a protein, it might produce a protein you might measure, and then you might say, well, it regulates a gene. But it's not really, it doesn't lend itself to this sort of ease in practice. It's a very hard thing to actually integrate these models. So they're showing an example where, this is a very generic example. They're showing how they might interconnect. A metabolite might affect the expression of a gene as well. So these are the RNA squares here, and these are the proteins. So it was mistaken on that. But basically the idea is that things can regulate a gene. You have them coming in from different models. Now they're very hard. These models are very hard to implement and calibrate and get them to work well together. But you can draw these pictures and you can show how they fit together. So they go over they actually go over topological analysis, which is something we talked about in the GNNs group. We've talked about this in our group previously, maybe last year. I think we discussed this a bit. So people are, there's this move towards topological analysis. So there are a lot of things you can do with these networks once you establish them. You can look at the things like modularity and hierarchy, which are the structures that occur within networks. If you get groups of things that group together, that's modularity. if you get things that are sort of organized underneath one another, that's hierarchy. There are different ways that you can characterize structure with networks. But another thing we might want to do is use topological analysis, which is, they say a considerable amount of work in this field is based on topological analysis of biological networks. In this case, graph-based representations also play a fundamental role. The analysis of the topological properties of these graphs, such as degree distribution, clustering coefficient, shortest paths, or network motifs, can reveal crucial information from biological networks, including organization, robustness, and redundancy. So, we want to be able to analyze the topology of the network. And you can do that through network statistics and other things like that, but you can also actually analyze the structure of the network and the structure of the data, in fact, to form the network. And so that's where this topological analysis comes in. But there's also topological data analysis, which can be used to inform the formation of these networks. So, so that they really stress standardization. So if you are familiar with the systems biology markup language, this is really the standard of the community. This is, in order to sort of make models that people can use across the community, we need to have a common language. So one of the ways they've done this is to work on the systems biology markup language. And this is a standard that people use to build models for many different systems. And then they can share them with people, just like we share pieces of software. We can say, these are the specifications, and, you can just plug in your data, and it should, be in a standardized format. So these models up here are standardized in the sense that they have, we have symbols for different things. So if it's a receptor, it's this symbol. If it's a signaling molecule, it's this purple symbol. Proteins are green, light green. mRNA is dark green. Metabolites are this sort of off green, and so forth. And so you have this convention where you can have a legend, and it tells you what these things are. And then, of course, if it's something that's common, you can also account for that in the standard. So, SBML is an XML-based language for representations of species, compartments, reactions, and their specific properties. So this is all chemical jargon, and this is kind of one of these things that they use, I think, a lot for metabolic networks. So they're focused largely on biochemistry here in this part. So SBML was initially focused on biochemical reaction networks. Therefore, it is not so well suited for modeling other kinds of processes, such as regulatory networks, which are better described by logical models. So even when you develop these standards, they become hard to map to other domains. So SBML is mostly biochemical in nature. It's hard to create models using that standard for regulatory networks. Regulatory networks, people generally use logical models, which means that when there's some criterion that's met, a gene is expressed, or it's expressed at a certain level. That's typically the standard. So people are developing standards for that, but that's a different standard. CellML is another one of these XML-based standards. This is a more generic form of an XML standard, and it's often used to model other types of networks, creating these graphical models with a common notation. And so they go over a number of different networks here, different approaches, process algebras, Petri nets. They show some formalisms with a visual representation here. So these are Boolean networks here. These are Bayesian networks. These are Petri nets. D are agent-based models. G are, let me see. G are cellular automata, of course. F are rule-based models. And then E is, they're just kind of transition models of some type. Interacting state machines. So you can see there's a diversity of models you can use for this. But the key is like bringing these all together for different processes that are related. So that's all I'm going to talk about with this paper. I think it's, if you want to know more, you can read about it. But it's a nice review of that area. And I think it's useful here because we talk a lot about these different things. And at some point, we want to merge them together and bring everything together in a way that we can interpret for the whole organism. It's to note that, like in the OpenWorm Foundation, we have a lot of parallel projects focused on like movement and focused on, we don't really focus on gene expression or metabolism very much, but we do a lot of stuff with, bioelectricity and things like that. So, but we don't really have a method to unify everything. We kind of have these parallel approaches, and they kind of fit together. You can use NeuroML to fit some of it together. A lot of the stuff from the brain, but it's hard to really get that standard and bring everything together at different scales and different systems across the body. So I'm going to get into something now called pushing, pulling, and non-reciprocity too. And so last week on the video, if you go back and watch it, I talked about pushing, pulling, and non-reciprocity. And it was this set of papers. One of the people was Amy Shire, who did some work on looking at morphogenesis as this process of pushing and pulling. And they compared it directly to the reaction diffusion model of Turing. So the reaction diffusion model of Turing, if we go back to this poster, this kind of pattern formation where you have basically something that's random and then it forms like dots or stripes or something like that. That is Turing reaction diffusion. This is a process where chemical morphogens interact and form these gradients, and then they form these clumps. Okay, and this is a chemical process where the chemical different species of chemical or different morphogen types kind of interact and form a boundary between one another. And so this is why you get this striping or this spotted patterning. In this alternate model that's been proposed, that's actually not what happens. What you have is these forces that occur in a sheet of cells, and they force like these buckles in the surface. And then those buckles are reinforced by some sort of activity. So it could be like developmental genes that are expressed in these certain areas where the buckles occur, but not other areas. So you get these buckles that occur in different parts of the, so they were using like an epithelial sheet as an example. And so this is epithelium that has like cells that are basically homogeneous. That sheet, whatever, these epithelial cells form a sheet. The sheet buckles, and buckling is not a uniform process. It forms these, different hills and valleys. And then those areas that are uplifted, as it were, they start to get reinforced by gene expression, developmental gene expression of different factors. And then you end up with these spots or these ridges or whatever it is, the pattern that you observe. So it's a very different way of viewing morphogenesis. And so these are some papers that kind of go in the same direction. So this is another paper that they did. I don't think I talked about this paper, but this is from this group. And their paper is called Emergent Cellular Self-Organization and Mechanosensation. Initiate follicle pattern in the avian skin. So they were actually looking at the avian model. They were looking at skin. They were actually looking at dermal formations. So they were looking at where the follicles will be for feathers. So the follicles are points on the skin where the feathers will grow out, but the follicles need to form, and they're different from the rest of the skin. So you had this buckling process, and you ended up with going from like sort of a homogeneous field to this inhomogeneous field where you have these spots that are going to become something, in this case, follicles. So the abstract of this paper reads, The spacing of hair in mammals and feathers in birds is one of the most apparent morphological features of the skin. This pattern arises when uniform fields of progenitor cells, and these are these epithelial cells, the epithelial progenitor cells, diversify their molecular fate while adopting higher order structure. So diversifying their molecular fate means that certain things are expressed in some cells that experience this buckling force versus cells that do not. Using the nascent skin of the developing chicken embryo as a model system, we find that morphological and molecular symmetries are simultaneously broken by an emergent process of cellular self-organization. The key initiators of heterogeneity are dermal progenitors, which spontaneously aggregate through contractility-driven cellular pulling. So this is this buckling I'm talking about. It's this contractile force. It's pulling at the cell, the sheet of cells, and it's forming these places where you can get these spots. Concurrently, this dermal cell aggregation triggers the mechanosensitive activation of beta-catenin in adjacent epidermal cells, initiating the follicle gene expression program. So we have this force that's initiating this gene expression cascade. It's like one factor that's really driving this, but it's driving this. It turns this program on, basically. Taken together, this mechanism provides a means of integrating mechanical and molecular perspectives of organ formation. So this is some of the feather primordium formation that they show in some figures. These are stains where they're like DAPI stains and Echidheron stains where they're looking at the surface from day 6 to day 8. You can see like you get one of these dermal rises, as it were. You can see these, and then you have BMP2, which is something that's being expressed as an example of how this program is turned on in these locations. So BMP2 is being turned on in this image. You have the DAPI stain, which is this. You have DAPI and Echidheron. Actually, you have beta-catenin here. So beta-catenin and DAPI together in this image, and then this is the DAPI, some laminar marker, and then Echidheron. So this shows the different things that are being expressed in this process over like from day 6 to day 8. It's a very tight window where this happens. In this day 6 of day 8 of chick development, which I don't know what the point is, but it's probably mid development or something like that. It's not very far along. So yeah, this is another example here where you have a control versus something that's been treated. The treatment actually, so yeah, this XAV939, this actually I think inhibits the formation of these sharp boundaries. So it's like something they can do like in a culture. They have a control, and then they have this, and it's inhibiting the formation of these sharp boundaries. And you can see here that this is shown in the stain, where this is the result of inhibiting boundary formation. So that they're able to do this for just within the chick embryo. They're able to show they in these kind of papers they show all these different possibilities. They like to stain the sample quite a bit and have these different things that are expressed and show images of it, because it's really kind of they'll have quantities, but they'll always show the images. So so it goes through that, and then it kind of goes through puts it on different substrates, and I think this is in a culture, stiff substrate to flexible substrate. So as you go along, the substrate makes a difference. There's low traction and high traction, so they show some differences there. So they want to show kind of the context where you get this sort of pattern formation. And so if you're putting these samples of the tissue on gel, you take them out of the embryo and you put them on a gel, and the gel is of different sort of properties of the gel. So it's stiff versus flexible. It allows the cells low traction versus high traction. You get these differences. So the substrate, or where the sort of the surface on which this occurs, matters. And so there's a surface in the embryo. The skin is just one part of it. It's forming on other parts of the embryo. So there are forces at work here that are this process requires a specific physical context. And then this is the movement of beta-catenin into the nucleus and the forming primordium. This is mechanically triggered and upstream of the primordium gene expression program. So this is where you get these different, like, this is one example. You have this gradient of traction, you have this gradient of resistance, and if you go to different points on these continua, you get these different effects. So you get clustering or you get dissociation. And so you can see that the dermis compresses epidermis. So the dermis is, I think, on top, the epidermis is here, and it's compressing the epidermis. This force of the dermis compressing the epidermis causes translocation of beta-catenin. And so you can see this in the cross-section, that this, as this epidermis actually is on top, dermis is on bottom. Epidermis starts to buckle, it compresses the dermis, and then the dermis has, there's some, this creates forces within the dermis, and you start to get this pattern formation. So pattern formation accelerates as these forces are introduced. So that's a very interesting approach. There's some other papers, though, that are related to this. This is another paper on growth and form of the gut, and this kind of goes through some aspects of gut formation in the chick embryo. This is another example of physical forces playing a role in development. So in this paper, the developing vertebrate gut tube forms a reproducible looped pattern as it grows into the body cavity. Here we use experiments to eliminate alternative models and show that gut looping morphogenesis is driven by the homogeneous and isotropic forces that arise from the relative growth between gut tube and the anchoring dorsal mesenteric sheet. Tissues that grow at different rates. A simple physical mimic using a differentially strained composite of a pliable rubber tube and a soft latex sheet is consistent with this mechanism and produces similar patterns. So you can actually produce this sort of morphogenetic pattern using physical analogs, which is interesting. And so they built a mathematical model. The predictions of their theory are quantitatively consistent with observations of intestinal loops at different stages in the development of the chick embryo. So do they have pictures of this loop? This is the mathematical model. I'm kind of hoping for some pictures. I don't know if they have them. Sometimes they have them at the end. Here we go. So this is the rubber simulacrum of gut looping morphogenesis. So this is our gut looping morphogenesis here, where we have like almost like this ribbon. You can see the looping. And so this is this blue and this red. These are basically a very similar thing to what we saw in the other paper. You get these things that form layers, one on top of the other. There's some stretching and relaxing, and you get this patterning, this looping formation. And it's affecting the tissue underneath it as well. So to construct a rubber model of looping, a thin rubber sheet is stretched uniformly across its length and then stitched to a straight unstretched rubber tube along its boundary. The differential strain mimics the differential growth of the two tissues. So these tissues are growing at different rates. The system is then allowed to relax free of any external forces. And so then upon relaxation, the composite rubber model deforms into a structure very similar to the chick gut, which is this sort of layering, this sort of image. This is morphometric and mechanical measurements of chick gut. So this is, again, this looping. They're able to do some quantification of the model and make some predictions. And then this is more of this modeling where they can make predictions for loop shape, size, and number at 3 stages in chick gut development. So they're able to do this for different stages of development. And then finally, this is an example of chick at E12, quail at E12. And E12 is just the developmental day, finch and mouse. So you can see this in different species where you can make comparative predictions for looping parameters. So this actually happens in a wide range of guts, and they're able to predict for each of these. They use the chick model to develop this technique or this model, and then they apply it to other species. So that's a nice example of this in sort of a more mathematical context. This paper, of course, also talks about gut villi. It's called vilification. And so this is by a group of people, including Amy Shire, some other people in here, and they kind of go in the same direction. So the abstract of this paper reads, the villi of the human and chick gut are formed in similar stepwise progressions, where in the mesenchyme an attached epithelium first fold into longitudinal ridges, then a zigzag pattern, then finally individual villi. We find that these steps of vilification depend upon the sequential differentiation of distinct smooth muscle layers of the gut. So you see the same process as the looping. You have these different layers. They're growing at different rates. They're changing their shape in different ways, and then the change in shape in one layer is affecting the shape in other layers. This sort of the sequential differentiation leads to a restriction of the expansion of the growing endoderm and mesenchyme, which are the other layers, generating compressive stresses that lead to their buckling and folding. So in this paper, they introduce a quantitative computational model incorporating measured properties of the developing gut, and it matches the morphogenetic or morphological patterns seen during vilification in a variety of species. And so then finally, we have this paper on self-assembly of tessellated tissue sheets by expansion and collision. This is a different group of people. And this abstract reads, tissues do not exist in isolation. They interact with other tissues within and across organs. While cell-cell interactions have been intensely investigated, less is known about tissue-tissue interactions. So cell-cell interactions are when cells are behaving, interacting with one another. We also have tissue-tissue interactions, which are, of course, very important, as we've just mentioned. Here we studied collisions between monolayer tissues, which are things that form a single layer, with different geometries, cell densities, and cell types. So now they're not interested in the interactions between layers per se. They're interested in monolayer tissues that have different shapes and different densities of cells and types of cells, which can actually act very much like this multi-layered structure. First, we determine rules for tissue shape changes during binary collisions. So they take cells one at a time, they collide them. They describe complex cell migration at tri-tissue boundaries. So if you have different tissues that are coming together, you can describe the sort of how cells migrate in that boundary space. Next, we propose that genetically identical tissues displace each other based on pressure gradients. So these should be all identical tissues. They shouldn't have any, genetic mutations between them. Displace each other based on pressure gradients, which are directly linked to gradients in cell density. So this is, again, we're dealing with gradients. We're not dealing with chemical gradients. We're dealing with physical gradients. We present a physical model of tissue interactions that allow us to estimate the bulk modulus, and this is a measurement of deformity in tissues and other types of things. I know Susan knows this pretty well. Bulk modulus of tissues from collision dynamics. Finally, we introduce Tessellate, which is a design tool for self-assembling complex tessellations from arrays of many tissues. We use cell sheet engineering techniques to transfer these composite tissues like cellular films. So tessellations are like different tilings of usually in like mathematics, they use the term tessellation to talk about how you can pack different shapes into a space, a continuous space. And, there are different ways you can tessellate a surface. You have like triangles or circles or whatever. In this case, they're using cells to tessellate this tissue. So cells are packed into this tissue in different ways. And so this is actually a very interesting approach. And they're actually advocating for, so this is the shape of colliding tissues. And these shapes are stereotyped and predictable. So when you get two rods, for example, that collide, they form this sort of composite. When you get two circles that collide, or two like blobs, they form this, like it looks like an ice cream cone almost. And the same thing with these blobs of different sizes. It almost looks like a shoe print or a light bulb. And then if you have a long rod and a blob, they come together and they form this shape. And it actually looks like something that's from the embryo. I mean, they're all from, I guess, an embryo, but they have these distinct shapes and they're predictable. That's what they're saying here. So, I'm not going to get too much into this paper. I'm just going to point out they did a lot of like engineering type analysis, and it's really, interesting work. It kind of follows up very nicely with some of the things we've talked about. So these are tessellations, and again, these are shapes that are like packed into a surface. So this is this type of thing here is a target tricolor tessellation, and this just has like 3 different colors and 3 different sort of sides. This looks like these cubes that are packed into the space. This is another example of a tessellation where you have a repeating pattern, but they're packed in, so it's continuous. There are no gaps in between them. This is another example of a tessellation where you combine these blobs into a continuous form, and so on and so forth. So that's I wanted to follow up on that topic. And I think that was a pretty good representative sample of what's going on with a lot of this work. So we have one comment here. This is a great overview. Thank you, Morgan. Did we have any questions about that or comments?
Just that the bulk modulus is mostly due to water in cells. It dominates over the elasticity and viscosity. So one has to be careful when measuring that.
Yeah, I think a lot of these are very controlled experiments, or some of them are controlled experiments. I think, some of the things that you measure in culture maybe don't map to the organism if you actually do things in vivo, which is to do it in the organism and observe the thing in the organism. So, I mean, there's a lot of water in this too. You're right. It's, there's water everywhere there's water in the cell, there's water maybe outside the cell. So it's a very,
just to add to that that water can also take different forms when it interacts with biological structures, and I'm just thinking clathrates, and I think there's some other papers that suggest that that the water should be modeled, or we should be careful in our assumptions about movement inside the cell. I mean, I'd love to hear someone who's got more knowledge on this speak, but I think it's really interesting that water can actually behave differently, and we're not just talking about freezing.
Yeah, I mean, and people, I think they've done this, but they do it in separate papers, so you don't get they don't bring it in. Sometimes these modeling papers or these papers where they do experiments, they're careful what they test. So they're doing this very focused study, but sometimes there are a lot of things that are affecting, and I talked earlier about the systems biology models. That sort of thing where you not only have these different sort of processes like gene expression or proteomics, but you also have these things like physics and water and things that are pretty standard in biology, but they don't have a model for them. They don't fit into these things.
Now, the cell is a very packed environment.
Yeah.
Like it's, you get condensations in cells. I think you had an article about that.
Yeah, I think at one point.
Previously. Yeah. Anyway, you actually get condensates in cells because it's so packed.
Right.
Packed with proteins, etc.
Yeah.
Yeah.
Yeah. So did anyone else have anything before we go? You want to bring up anything?
I also would like to know more about Petri nets.
Oh yeah.
I've tried to read up on this, and they do seem like a strange hybrid of some things that I just not quite wrap my head around yet.
Yeah, it's like Petri nets and reservoir networks. Those are two things that are really kind of like I can't really wrap my head around fully. But
yeah. That reminds me of a reservoir network paper that I'll try and post in Slack. I'm just did the school run, so I'm still in the car.
okay,
But an interesting reservoir paper on hopefully we can talk about. Yeah, that'd be great.
Okay, so thanks for attending. Next week we'll have more updates on GSoC and more papers and more discussion. So have a good week.
Bye.
Bye.
Take care.
Take care.